Hi, everyone. All right, we're moving on to chapter 15. Couple of notes as we begin to move ahead. First of all, your exam, guys, I have looked at the results of the poll. It will be a 90-minute Blackboard exam. I'm very glad that's what you guys chose. I think it's what's best for you, and definitely what's best for me too. So so it is going to be the same format. It is going to be the same sort of multiple choice breakdown. It's going to be worth the same point values. You will be having a little bit longer than you would normally have had in class, right? In class, we have like a 50-minute class. I always give you guys a little extra time if you need it. But I'm going to go ahead and time it for 90 minutes. It will be on April 14th, or sorry, nope, April 17th, don't panic. And what I'm going to do is I'll open it at midnight. I'll close it at 11:59 p.m. It'll just be set to run for 90 minutes. Once you open it, you must finish it. There will be no pausing it, okay? But what I will allow is for you to move back and forth within the exam. Let me tell you a little pro tip from me to you about taking these exams on Blackboard. I am not naive. I recognize that you're at home and you have resources at your disposal. And so you're going to want to look things up. And I am not going to prohibit that because there's no way for me to police that. But let me caution you against something. One of the things that we are finding since we've moved online is that students are actually, even despite us giving them more time, despite us keeping like the length of our exams the same and giving them additional time to do it, students are struggling to finish exams on time. And our guess is that what's happening is very little preparation is happening prior to the exam, and students think that they'll just look up every single answer. That is not going to give you enough time to do that. 90 minutes is not sufficient for you to look up every single answer. So my recommendation to you is that when I give you the exam next Friday, that you take it, you go through it, you answer the questions that you feel good about, and then you leave yourself a small subset of answers that are, a small subset of questions that you want to look up and verify the answer to. If you try to look up every single question, you will not finish in time. So an online exam does not mean that you shouldn't study. Here is my plan for the exam moving forward. We are going to have it cover chapter 13, chapter 14, chapter 15, which today will be composed of a single lecture. As we move forward, I'm trying to condense these chapters down a little bit, hit the high points, so I can still cover some of the material I want you to know about. So that means that on Monday, I'm actually going to give you either 2 short lectures or 1 longer lecture that combines chapters 16 and 17, all right? So you're not going to have the entirety of these chapters. I'm only going to hold you responsible for the bits that I discuss in the lecture. That'll be on Monday, so let me go ahead and put that there, that will be posted. Then Wednesday, when we would have ordinarily had class, I will not be posting new lectures. That is just going to be your time to catch up and study, okay? And then Friday will be your exam. Now, a couple other things I want to mention. Monday is also a big day. You've got some homework due and you have a lab due. And as I've said to a couple people, if there is some reason that that due date is giving you trouble, then just let me know. I can certainly be flexible, but I would prefer if you contacted me ahead of time. Similarly, if for some reason there is something in your life on Friday that you just, I don't know, maybe you have a sibling that is going to be online all day. I don't know what your personal circumstances are. If Friday presents some kind of problem for you, then I would like to know ahead of time, okay? Again, I can be flexible, but I would prefer to have a little heads up. Other things on the agenda, Monday, I'll also be posting your next lab, but that's not due for 2 weeks, so don't worry about that right now. The last thing I want you to know is that your exam is Friday, April 17th. You are being asked to make a decision about the new grading scheme by Monday the 20th. I have already contacted my department chair to let them know that I'm going to be granting you an extension. I'm going to have your exams graded by Monday the 20th, and then I'm going to give you 24 hours to think on, based on your exam performance and your current average, whether you would like to switch to the new satisfactory, unsatisfactory grading scheme. And I'll be discussing the pros and cons of that grading scheme in another video, okay? I do have some information on it in this PowerPoint that I'll upload to Blackboard, but I'll actually go over it in a video later on. Okay, moving forward, let's talk about chapter 15. So chapter 15 is basically going to focus on translation. So we've gone from DNA to RNA at this point, and I always tell my freshman students, that's transcription. You're transcribing. When you transcribe something, you're remaining within the same language. You're making a copy of something in the same language, right? If you make a transcript of this video, you are just writing down what I've said. You're going from English to English. You're going from video to writing, but you're still going from English to English. And I think that's a nice analogy. Transcription is going from nucleic acid to nucleic acid. But when we move from RNA to protein, which is what we're going to be talking about today, you are translating something. You're going from the language of nucleic acid to the language of amino acid, right? So this is going to be our translation. Okay, so that's what we're focusing on in chapter 15. And so in thinking about that, I did want to do a little bit of review about amino acids themselves. We talk so much about nucleic acids, but let's talk a little bit about amino acids. And I'm going to erase this because hopefully this is all freshman biology for you guys. I need some room to write. Okay, in thinking about the basics of an amino acid, let's kind of look at their chemical structure the same way we looked at structures of nucleotides. And amino acids all share a very similar structure. So you've got a central carbon against which you have bonded a hydrogen, a functional group called a carboxyl group, and an amino group, okay? And this basic structure is what IDs amino acids as amino acids. But just like in nucleotides, you had different functional bases. You had A's and C's and T's and G's. Instead of having bases, we have in amino acids what we call the R group, these side chains. And so I have for you here a list of the 20 amino acids that we're going to be concerned with in translation. And I am not interested in you memorizing these different R groups. But what I want you to know is that the R groups are what give the amino acids their chemical properties. Okay? So let me give you a for example. Let's focus in on lysine. We've talked about lysine a little bit because we talked about it with histone proteins. Lysine is positively charged. Remember that? And if you start adding things to the lysines and you neutralize them, then you neutralize the charge on the histone proteins, and the DNA doesn't stick so well to those neutral histones, right? So what gives lysine its positive charge is this functional R group right here, okay? Similarly, arginine is another positively charged amino acid, and that positive charge comes from its R group, okay? We can look at another set. So if we look at negatively charged amino acids like aspartate and glutamate, you can see that it's their functional groups here that are giving them a negative charge. And so you can look through and see that each of these different amino acids all have different R groups, and that's what determines how they function. Now, in thinking about translation, I told you that we're going from RNA, of course. We're going to amino acids. We're going to a sequence of amino acids. So if you think of it as beads on a string like this, right? Those are all individual amino acids, or this is another way of looking at that. That is a polypeptide chain. That is not a functional protein, okay? What do I mean by that? Well, at this point, just a string of amino acids doesn't really do anything. But what happens is after we translate the RNA into amino acids and we make what's called the primary structure, which is just a sequence of amino acids, the R groups are going to start to interact with each other, and they will form a secondary structure. And the two secondary structures that we find in amino acids or polypeptide chains, you may remember from Biology 105, they are called alpha helixes, or helices, it's actually the plural. You have an alpha helix or a beta sheet. Sometimes they're called beta pleated sheets. If you've ever like taken a piece of paper when you were a little kid and folded it up like this to make a fan, but it was still a flat sheet of paper with some kinks in it, that's kind of what a beta sheet looks like. So that's the secondary structure. And it's not until we get to this tertiary structure here that we see a functional protein. Okay? Oops. So what happens is that the secondary structure knots up on itself again, and it's going to give a three-dimensional shape to the protein so that maybe if it's an enzyme, it'll have an active site, or if it's a structural protein like collagen, it'll be the right shape to give some structure. This example I have for you here is part of a subunit of hemoglobin, and this tertiary structure has given it a three-dimensional shape so that it can accept what's called a heme group, which is the iron-carrying group in hemoglobin. That is a heme. Okay, it carries the iron. And the reason your blood needs iron is because the iron binds to oxygen. Okay, so we now have a functional protein typically at the tertiary level, but there are some proteins that take it one step further and they go to what's called a quaternary structure. And that simply means that we have several individual proteins that form a multimeric structure. A multimer, which means many units, right? So you have 1, 2, 3, 4 individual proteins that come together to form a single sort of mega protein. And so this is hemoglobin is a great example of that. Ooh, okay, I ran out of room. You get where I'm going. So, you know, we talked about sickle cell anemia. It turns out that there's one subunit, the beta hemoglobin. I'm just going to put beta H. The beta hemoglobin subunit, so only one of them is messed up in sickle cell anemia and it doesn't bind oxygen properly, okay? So I just kind of wanted to review the different levels of protein structure. What we end up with as the end product during translation is actually here at the primary part, and then different folding and then processing can occur to confer these other functional structures upon the protein. Now, in thinking about making an amino acid sequence, remember that we're going, of course, from DNA to RNA to amino acids, right? So that means that the RNA, the DNA, and then the RNA, there must be some way that the base pairs in the DNA and RNA code for amino acids, right? Now, one of the things that you've probably been told is that there are 20 amino acids that go into 20 different varieties of amino acids that can go into making a protein, right? That's actually a wee bit of a fib. There's lots and lots of different amino acids, but there are 20 what we call proteinogenic amino acids. So there's 20 that are coded for in the genetic code. So Francis Crick, after he and his buddy Watson, who we decided was just a garbage dumpster fire of a human being, Francis Crick, after they devised the structure of DNA, the next question that he set himself to was, what is this code? How do the A's and the C's and the T's and the G's equal these various 20 amino acids? You've only got 4 bases, right? So you've only got 4 options, ACTG or ACUG if you're RNA. So like if an A coded for an amino acid, or a C coded for an amino acid, or a U or a G, that would only mean that you had enough, if you had 4 bases, I'll put bases here. If each individual base codes for an amino acid, then you would only have 4 amino acids that you had a code for. So they knew that couldn't be the case. So then they said, well, what if you had 2 amino acids? What if like AU was 1 amino acid, AA was another amino acid, AC, you get where I'm going. Well, mathematically, that works out to be 4 to the second possible combinations. And if you work that math out, that's 16 possible combinations you can come up with just trying to make combinations of 2 different amino acids. I'm sorry, 2 different bases, right? You come up with 16 combinations just pairing up 2 different bases at a time. Well, we need at least 20 unique combinations. So pretty quickly, people figured out, Francis Crick among them, figured out that if you have a series of 3 bases that make up a code, like AUG, AAC, ACU, etc. If you have 3 of them at a time, you can come up with 64 possible combinations, which is more than enough to code for the 20 amino acids that we see in proteins. So this is what we call the triplet code. And each triplet, I hope you remember from general biology, is called a codon. Oops. Okay, so here, this is a codon chart, and you usually see these in terms of the RNA sequence, okay? So you've got UUU, UUC. These are all individual codons that code for the amino acids that you see indicated here. So like this is phenylalanine, leucine. Leucine has a lot, isoleucine, methionine, valine, etc. Okay? So that was the first thing that they figured out about the code. And it turns out there are 3 features of the genetic code that I want you to really understand. The first is that the genetic code is what we call degenerate. Now, I don't mean degenerate in the form of being like a lazy, bad person. Degenerate, is it erate? Hold on. Oh God, I'm a terrible speller. I hope that you guys will bear with me. Yeah, it's an E. Come on, Beth Jones. Degenerate. Thank you. Okay. Now, Francis Crick actually borrowed this terminology from quantum physics, which means multiple states having the same meaning in quantum physics. And you can immediately see how he saw this as analogous, because you've got multiple here. You've got, look at leucine. You've got 1, 2, 3, 4, 5, 6 different codons that code for leucine, because we've got 64 possible combinations. So it turns out that there are 3 codons that are what we call stop codons. They do not code for amino acids, so they are not correlated with amino acids. Then that means that we have 61 others that code for amino acids. All right. And so there are some amino acids that have lots and lots of different codons that are associated with them. And so if a set of codons codes for the same amino acids like you see here with leucine, we call these synonymous, like synonym in English, synonymous, I can do this, synonymous codons, okay? In other words, different words that have the same meaning, right? Okay, now, one question that you may be asking yourself, or that hopefully you remember from general biology is, okay, so we've got these codes. How do these codes get turned into amino acids? How do we associate an amino acid with the code? And that answer resides in one of the RNA classes that we've mentioned previously, tRNA. So let's look a little bit at the structure of a tRNA. It is, you've probably seen it before. It's characterized by this shape where you have 3 hairpin structures, 1, 2, 3. And at the bottom here, you have what's called an anticodon. So let's just say that the codon here, and I'm going to use one, UCU. I don't have these memorized. I just planned some of these examples. UCU is a codon that codes for serine. Okay, so serine's an amino acid. Well, the anticodon is the complementary pair to that. So that would be AGA. So this would pair with, on an mRNA strand, just like here, you have an anticodon of CUC. It's going to pair with this codon of GAG here. And what you have at the opposite end of this tRNA is the amino acid that goes with this codon. So a tRNA that has an AGA anticodon matches with the codon for serine and has a serine at its very tippy top. And that's how you're going to match the amino acids with the mRNA molecule. Now, something interesting about tRNAs is depending on what kind of critter you are, you have 30 to 50 tRNA types. But remember, we have 61 codons that code for different amino acids. So there's not enough tRNAs to code for all 61 codons. And let me give you an example. So in serine again, it turns out there are 4 different codons that are serine, right? UCU, UCC, UCA, and UCG. All of these bad boys code for serine. These are all synonymous codons. But most of us only have 2 tRNAs that code, that give us a serine, okay? How does that work? Well, there's a phenomenon called tRNA wobble. All right, and what happens in tRNA wobble is that the transfer RNA, if you notice, these all have UC at the beginning. So the transfer RNA binds really well with the first base pair, and the anticodon binds really well with the second base pair, but the third base pair, not so much. So for example, we have a tRNA that will bind with the C or the U, okay, weakly. Binds really strongly here, but it binds with these two weakly, so it wobbles between these two. We have another tRNA that binds the U and the C really well, and the A and the G wobbles a bit. It binds, but it binds weakly, okay? Any tRNA that you have that can accept like different letters at the end of the codon is called an iso-accepting tRNA. Oof, okay, so now we know about tRNAs and we know about proteins. We have enough information to begin thinking about translation. Now, the first thing you need to know about translation, oh, sorry, we were talking about genetic code. Let me go back. So I just told you about degeneracy of genetic code, and that's what we were talking about when we were talking about the multiple codons, and we were talking about the different tRNA types. The next thing about the genetic code, sorry, I got ahead of myself. So we're still on the genetic code. The next feature I want you to know, so this is number 2. Number 1 was degeneracy. Number 2 is that the code is non-overlapping. This was a question that researchers were really grappling with when they were trying to figure out the code, was did the code happen in consecutive groups of 3 like this, or did it overlap like you see here, okay? And it turns out that it's consecutive groups of 3. It is non-overlapping. It begins with a start codon. Start codon is AUG. That actually codes for the amino acid methionine. So all amino acid polypeptide chains begin with methionine. Now, sometimes this initial methionine is cut off later, but every single polypeptide chain is going to have this AUG methionine start codon. And then they all end with stop codons. So let's see here. Those can be UAA, UAG, or UGA. These are all your stops. And remember how I said that they don't code for amino acids? That's because there are no tRNAs, oops, with anticodons that correspond to these. There are no tRNAs for the stop codons. So 1, the code is degenerate. 2, it is non-overlapping. And the very last thing I want you to know about the genetic code is number 3, it is universal. It doesn't matter if you are a bacterium, if you are an archaea, if you are a Mycoplasma capricolum, which is a fungus, or if you're a human. The code, if you are, when I say AUG is the start codon, it's that way for everybody. Now, in recent years, we found a few exceptions to the universality of the code, as you can see here in this table, but by and large, the general rule is that the codons code for the same amino acids regardless of what kind of critter you be. All right? So those are the three things. Now we're ready to move on to translation, okay? So again, I put this up here. This is just an overview of what translation is going to be. And I wanted to kind of familiarize you with some of the diagrams that we're going to be looking at. So these rectangles are going to signify our messenger RNA molecules. You're always going to be reading these messenger RNA molecules from the 5' to the 3' end of the molecule, okay? Ribosomes are going to look like little hamburgers, and they're made up of 2 subunits. Now, ribosomes of prokaryotes and eukaryotes have some differences, but they both have a small subunit and they both have a large subunit, okay? So they both have kind of this hamburger configuration that's going to sandwich the mRNA between them, okay? This is our start codon here, our AUG. And what's going to happen is this ribosome is going to move from the 5' to the 3' end of the mRNA molecule, reading 3 base pairs at a time to get a polypeptide chain out of all of it. Now we can break translation down into 4 different phases, and these should look really familiar. Anytime you're making a polymer of some kind in a cellular environment, you're going to have an initiation, an elongation, and a termination phase. So we've seen that with DNA replication. We've seen that with transcription. We're going to see it again with translation. All right. The only thing we're adding right here is something called tRNA charging. What is that? Well, it's pretty simple. Basically, in thinking about tRNA charging, tRNAs, they all have these different anticodons, but otherwise they're really similar in their structure, and they all end in the same sequence, the CCA sequence. So how the heck, and they're floating around in your cell without amino acids attached to them, okay? So there's no amino acids. And the act of tRNA charging is just the act of adding the amino acid to the tRNA, okay? That's all it is, all right? But if all tRNAs end in this same end sequence, how the heck do you know which one needs an adenine, or sorry, which one needs a serine put on it, which one needs an isoleucine put on it, blah, 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 blah. Well, that work is done by a series of enzymes called aminoacyl tRNA synthases, sorry. And they basically are able to determine which tRNAs get which amino acids stuck to them. And it turns out that there are 20 different of these enzyme synthases. I think this should be an A. Okay, there are 20 different aminoacyl tRNA synthases in a cell, one for each type of amino acid, and they're able to recognize which tRNAs get which amino acids. So that's the first step, is simply adding the amino acids to the tRNAs. The next step is getting your messenger RNA molecule, which is the template for how you're going to make this polypeptide chain. We're going to have to get it to the ribosome. Remember that the ribosome is the unit of protein synthesis. Now, if you are a prokaryotic critter, that's fairly easily done because there's no nucleus, right? There's no nucleus there. So after the DNA is transcribed into RNA, the messenger RNA then can associate with the ribosome right there. And what happens, if you recall, we talked a little bit about mature mRNA molecules. I told you that in the 5' untranslated region of a prokaryotic mRNA, there's a sequence that actually is complementary to ribosomal RNA. And it's called the Shine-Delgarno sequence, okay? It's got a name. So here you can see what's happening is the mRNA is binding to the small subunit of the ribosome because it matches, the sequence matches some of the ribosomal RNA. So in order for translation to begin in a prokaryote, the first thing that has to happen is this mRNA complementarily base pairs with the ribosomal RNA to associate. Now, you may be asking yourself what this little protein is here. I'm not holding you responsible for it, but what that does is IF3, basically by binding to the small subunit of the ribosome, it keeps the top bun of the hamburger from smushing down. It allows time for this mRNA to line up and bind without the top part coming and trying to bind, okay? Now, after we have the mRNA in place, the next thing that's going to happen is we have a start codon. And I told you that start codon codes for methionine. And so in bacteria, they actually use a little bit of a modified version of methionine called N-formal methionine, but you don't have to worry about that. In eukaryotes, it's just methionine. I don't care if you know that or not. Basically, the methionine tRNA comes along and it binds through complementary base pair rules. You've got the anticodon here, you got the codon here, you got methionine here. At this point, there's some other proteins that join the party. These other proteins that join the party promote the top bun of the hamburger coming along and clamping down on this entire apparatus. We now have everything in place to begin translation, okay? We have the template, we have the ribosome, and we have our very first tRNA. So the order of operations is the mRNA binds, that's one. The start codon / methionine tRNA binds, that's step 2. And then the large subunit of the ribosome binds, that's step 3. Once you have all of those pieces in place, you can move on to the next phase, which is going to be elongation. Now, this is how things happen in prokaryotes. Of course, it's always going to be a little bit more complex in eukaryotes, all right? We're not going to go through all of the various details involved in initiation in eukaryotes, but there's a couple things I wanted to mention. If you recall from your lecture on chapter 14, I told you that a mature mRNA in a eukaryote has a 5' UTR, untranslated region, a 3' UTR, got the coding sequence, which I said was the dumbest abbreviation in the world, and it has a 5' cap. And I told you that 5' cap was going to be important for translation. Well, what the 5' cap does is basically there are a series of proteins called cap binding proteins, and they form the cap binding complex. Or CBC, okay? And so the cap binding complex in the nucleus, after all of this has been processed through the mechanisms we talked about last lecture, these cap binding complexes bind here, and they promote export of the mRNA from the nucleus, and they also help the mRNA to bind to the small subunit of the ribosome, okay? They help it to bind. Then remember how I told you the poly A tail was also involved? What's gonna then happen is another set of proteins bind to the poly A tail. They then help other proteins come along and put the top bun of the hamburger on top of all of that. And then elongation can proceed. Once you have the entire ribosome hamburger sandwiching the mRNA burger patty, if you will, if we're gonna stick with our analogy, then you can begin the process of elongation, okay? Now, elongation is actually the simplest part of this process. To understand elongation, what I need you to understand is that there are 3 sites in the ribosomal hamburger, okay? So if I draw the ribosomal hamburger here, I'm gonna try to draw it with my pen. Here's the top bun or the large subunit. Here's the bottom bun or the small subunit. We have our mRNA going this way, right? Well, there are 3 sites that connect that have spaces for tRNA molecules. The first site is called the aminoacyl site, okay, right here. And that's the A site. The next site is the peptidyl site, okay. That's the P site right here. And then the third site is actually going to be an exit site where the tRNA will leave the whole complex, okay? So that's what you really need to understand how elongation will work. So let me show you some diagrams from your book. It's pretty simple. So let's go back to our example where we had in a prokaryotic situation, we had our first methionine bound. We had all that happen. We had, let's go back to initiation. So basically we had the small subunit and then the mRNA bound because it matched some of the RNA in the ribosome. And then our methionine molecule came along and it found its start codon. And then the third step was the top bun of the hamburger got put on. And when that happens, the position that this methionine is occupying is that middle seat, that peptidyl site, okay? The next thing that's going to happen is a new tRNA is going to come along and it's going to use complementary base pair rules to match its anticodon with the codon. And it's going to enter this A site. That requires a little bit of energy. So you see we're going to have GTP involved and it's going to be eventually be GDP. It's going to lose a phosphate group. So we're going to break a phosphate bond in order to insert this into our ribosome. So here we are in a situation where we have the peptidyl site occupied and the amino acyl site occupied, all right? The next thing that happens is the ribosome, through a dehydration synthesis reaction, is going to form a peptide bond here between the amino acids. See how they're right next to each other? They're just ripe for a chemical bond to form. So they are going to form a bond. And then like a conveyor belt, what's going to happen is the ribosome is going to move this direction, which is going to cause this tRNA to end up in the E site, this tRNA to end up in the P site, and it's going to open up an A site so that the whole sort of conveyor belt can operate again, all right? And as usual, I think this is most easily envisioned through an animation. And this time I'm going to remember to share the screen. So let's see here, which screen is it I'm trying to share? It is this one. This animation is from your book like the others are, and it is enabled for you. All right, looks like I'm on the right screen. Let's have a look.
In translation, amino acids are assembled into proteins. Bacterial translation is initiated in 3 steps. First, the small subunit of the ribosome binds to the mRNA so that the P site is positioned over the start codon. Second, the initiator tRNA with N-formylmethionine attached binds to the mRNA through base pairing between the codon and the anticodon. Third, the large subunit of the ribosome joins the complex to complete initiation. During elongation, amino acids are joined to a growing polypeptide chain. The first step of elongation is the delivery of tRNA and its amino acid to the A site. In the second step of elongation, a peptide bond forms between the amino acids. The third step of elongation is translocation, the movement of the ribosome down the mRNA. After translocation, the tRNA formerly in the P site is now in the E site, from which it moves to the cytoplasm. Now the elongation cycle repeats itself. A tRNA and its amino acid occupy the A site. A peptide bond forms, and the ribosome translocates to the next code.
I just wanted to point out, as this is leaving, can you see that this is an uncharged amino acid? So it's going to have to go through a step of charging before it can be reused in this process.
As the cycles of elongation continue, the polypeptide chain grows by the addition of amino acids to its carboxyl end. The last stage of translation, called termination, takes place when the ribosome reaches a termination codon. Release factors bind to the termination codon, causing the release of the polypeptide chain. The ribosome then translocates one last time. The tRNA and release factors exit the ribosome, and then the ribosome dissociates. Remember that several accessory proteins not shown here also participate in translation.
Okay, so hopefully that gives you some idea of how that all happens.
In translation, amino acids are assembled.
All right, let me go back to. Where is. There we go. Sorry, it took me a second to find where I was. Okay, back to this. So the last step was noted in the video is termination, and all I want you to know is exactly what you saw in the video, that basically once the ribosome reaches a stop codon, release factors come along, and then the entire thing dissociates, okay? The very last thing I want you to know from chapter 15 is simply that when this process occurs, and we see this in both prokaryotes and eukaryotes, but this is a prokaryotic example here, because what you see here is a situation where we have transcription, got DNA, and it's being turned into RNA, right? And so we're adding nucleotides at 3' end, and it's growing from at the 5' end, right? So it's definitely following the rules. Because there's no nucleus, ribosomes can come along and they can begin translation right away, okay? So they start translating right away, and they're going to, but they actually join from the 5' end and move toward the 3' end, okay? So what happens though is that you can actually have more than one ribosome on an mRNA at a time. And when you have that situation, it's called a polyribosome or a polysome. So for example, we have a ribosome here that must have started down here, and it's been trucking along and it's been translating. This one started down here, it's been trucking along and it's been translating. Same. So can you see, like, this one started down here, it's the furthest forward, it's gonna have the most complete polypeptide chain. This one will be shorter and shorter and shorter. But what this does is it increases the efficiency of translation. So you can have multiple ribosomes on a single mRNA, so you can pump out protein pretty quickly, okay? So as I said, I was gonna keep things from chapter 15 pretty short and sweet. I'll be posting 5 homework questions from this chapter. That'll make for a total of 30 homework questions that are due on Monday. Again, if you have any issues with my due dates, you just let me know and I'll make accommodations, and I'll talk to you soon. Bye-bye.